Medulloblastoma, as Dr. Scheineman mentioned, is a tumor of childhood. We rarely, if ever, see it in adults. And there really has to be some deeper information embedded within that. Why does this tumor only occur in a child or a developing human being? And so the nature of this cancer is that since it occurs in children, there could be some approaches, research approaches, that are better suited to study a more developmental tumor. And so today in this talk, I'm going to highlight some of the advances and some of the research approaches that have come from the field of developmental biology and developmental neurobiology, and tell you why it makes so much sense to take these approaches and apply them to the study of a cancer. And so, as you probably figured out, my lab, which specializes in brain tumors, takes a developmental neurobiology and stem cell biology approach and applies it to the study of brain cancer. And I'd like to sort of show you why taking things from two apparently very different boxes makes sense in the perspective of this childhood tumor. So the reason that I decided to become a scientist is because during my pediatric neurosurgery rotation at the Hospital for Sick Children, I met two little boys who were both named Christopher, and they were both 5 years old. And strangely enough, they both had the same tumor that Josh had medulloblastoma. And you can see this tumor here, as Dr. Scheineman described, in a part of the brain called the posterior fossa. It's here in the back of the brain. Here's the cerebellum in this case, which is squished by this very large mass which is taking up the MRI gadolinium, the contrast agent. It's squishing the brain stem, and instead of seeing the spinal fluid, the CSF, which is normally black, circulating nicely from the ventricles around the brain, down around the spinal axis, the CSF circulation is completely blocked here by this large mass, which is compressing the fourth ventricle and the aqueduct. And that results in this enlargement of the fluid space and the child presenting with symptoms of raised intracranial pressure, headache, vomiting, lethargy. So both Christophers came into emergency with very similar tumors. And they were diagnosed with medulloblastoma. And they were treated with our best possible gold standard therapy, which is to take the child immediately to the operating room and try to remove the tumor totally if possible, which was accomplished with both Christophers. They were both diagnosed with medulloblastoma by histology. As Dr. Scheineman said, the pathologist examines it under the microscope, and based on the cellular structure and some of the staining pattern as well of immunohistochemistry, the pathologist can categorize the tumor in one of those different 5 pathological subtypes. The most common 3 Dr. Scheineman was sort of showing you mostly. And in this case, both of the Christophers had a classic medulloblastoma, same pathology. So they went on to the same stratification of therapy that Dr. Scheineman showed you. They were both, actually in this case, they were both high risk, and they went on to chemotherapy and radiation. And yet one child died when his tumor recurred at 15 months post-therapy, and the other child was alive when I left my residency training and is going to university. So the question for me as a neurosurgeon in training at this point is, why? And in fact, that's the same question that you're going to hear from all of the parents and the patients themselves and the patients' families. Why? Why did my child die? Why did the other children survive? And it's very, very disheartening as a clinician to not have a good answer to that question. So there was a Greek philosopher named Heraclitus who said, "One cannot step twice in the same river." And the problem with studying any disease, cancer in particular, and something that is really now being very, very closely studied in cancer, is the fact that there's so much heterogeneity at every level of cancer. Heterogeneity meaning a mixture of everything. And so what we see is that at the patient level, there's a great deal of heterogeneity. Every patient is different, right? Then you have the tumor itself. Every tumor is different. They're different sizes, they're in slightly different places, they invade things differently. At a cellular level, although those tumors looked the same, they were obviously different, and they behaved different biologically. And now we find that when we study things at a cellular and molecular level, there the heterogeneity persists. So when you have this much heterogeneity in a tumor, how do you find a research approach that will address it, and that will be able to distinguish the differences, that will tell us somewhere embedded within the cells and within the molecules of those tumors, there's an answer about why one child would die and why one would survive? And that's really what we're trying to uncover. So it's like a very long detective tale or mystery that we're really trying to discover the answer to these puzzles. So the other problem, of course, is that cancer is a dynamic disease, and it's growing and it's changing every day. And some of what Dr. Scheinemann showed you, our typical and classical ways of addressing cancer, is we take snapshots over time of the patient. Here's an MRI, here's another MRI a year later, here's another MRI a year later. But in the meantime, if there's any cancer cells left in your body, they're growing and shifting and changing. How do we capture that? How do we capture that dynamic change? And so two of the problems actually that we're facing as scientists is how do we capture heterogeneity, and how do we capture something that's dynamic and changing? And hence, how do we portray or profile a cancer that is changing just like the waters that flow in a river? You can never step in the same river twice. So Dr. Scheinemann has already told you about medulloblastoma, but all I want to point out to you from this slide is that a lot of what we've learned from this disease comes from people who have familial or genetic cancer syndromes. And so I think a lot of the foundation of the information that we have learned comes from these syndromes. And only 1% to 2% of patients have a hereditary type of cancer where they have a genetic predisposition or some gene mutation that sets them up for this cancer. And in this case, there's two familial cancer syndromes that we've learned a lot about medulloblastoma from. One is called Garlin Syndrome, and I'm going to tell you a little bit more about that. In Garlin Syndrome, there's a molecular pathway called the sonic hedgehog pathway that is disrupted. And in Turcot Syndrome, there's a problem with another molecular signaling pathway called the Wnt signaling pathway. And something I want to point out to everyone is that the Wnt signaling pathway and the sonic hedgehog signaling pathway are incredibly important to development of all kinds of tissues and organs in our body. So aside from playing a role in cancer, they're very, very important in the development of normal tissues and organs. So Garlin syndrome is an autosomal dominant pediatric cancer syndrome where patients develop basal cell carcinoma as well as medulloblastoma, and they're exquisitely sensitive to ionizing radiation. This is a syndrome where the molecular biology is very similar to retinoblastoma. So everyone has two alleles or two copies of every gene. And with most of us, in order for cancer to develop, you have to have both copies of the gene knocked out and that signaling pathway disrupted in order for the cancer to declare itself. But in children who have a familial cancer syndrome, they have a germline mutation, so one copy is already disrupted when they're born. So they're actually a few steps closer to cancer than the rest of us, and that's why they tend to develop cancer at a younger age and usually a more severe form. So we learned a lot about medulloblastoma from Garlin syndrome because we found that these patients actually have a germline mutation in the patched gene, which is the receptor of the sonic hedgehog signaling molecule. In Turcot syndrome, of course, we found out there was a problem with the Wnt pathway. So I'm going to return to those things. So I'm going to show you the sonic hedgehog pathway to show you that, in fact, you need to know a lot about cell signaling to be able to understand this cancer. So I want to show you right here. Here's one cell, and here's another cell, okay? And the reason we call it cell signaling is because these cells are talking to each other. And the interesting thing about the hedgehog signaling pathway is that hedgehog is a small, soluble signaling molecule that is released from one cell. The signaling cell releases the hedgehog. Hedgehog diffuses over between the cells and binds to the receptor, which is on the other cell. This is the patched receptor. Patched normally represses this other receptor called smoothened, but when the hedgehog binds, this inhibition is released. Then what happens is further, these other signaling molecules are also released, allowing this transcription factor to go from the cytoplasm into the nucleus. This transcription factor, which is called GLI, binds to a whole bunch of target genes and activates processes involved in cell proliferation, cell growth. And this is how, if you have an overactivation of this pathway, you get too much transcription of the target genes that cause too much cell growth and cell proliferation. And the very interesting thing about this is that it's fitting that tumors that occur in childhood occur from dysregulation of these normal developmental signaling pathways. So as you're growing up from a baby into a child and your tissues are developing and so on, they're normally this process is regulated very tightly by sonic hedgehog and by other such signaling pathways. So the cerebellum, when it's developing during embryogenesis, actually what happens is you have these stem cells that arise in the roof of the fourth ventricle, right in that area where I showed you on the MRI the tumor was growing. growing. And those stem cells migrate over the surface of the cerebellum and turn into what we call external granule cell layer precursor cells. And in fact, this entire process of this migration, proliferation, and how they turn into those cells is regulated by sonic hedgehog. And in fact, the more sonic hedgehog you have, the more proliferation you get. And the very interesting thing is that the proliferation is regulated by hedgehog, which is secreted by one type of cerebellar cell within the 6 layers of the cerebellum called the Purkinje cell. So these guys release the hedgehog, just like I showed you in that signaling diagram, and then it diffuses down and binds to the patched receptors which occur on the granule cell precursors. So there's cells communicating in a normal way to regulate your cerebellum turning into a normal, healthy cerebellum. But what may happen during cancer is that, like I mentioned, you either have too much hedgehog or you have a patched receptor that is mutated and not able to receive the hedgehog signaling properly, and so instead that receptor is constitutively active or always active, then the end result is you get too much transcription of those genes regulating proliferation. And what is cancer? Cancer is really no more than a cell that grows inappropriately and proliferates too rapidly, that it can no longer be regulated by its surrounding tissues, right? So what we have here is 2 different ways, either a cell seeing too much hedgehog or a broken patched receptor. Either of those things can cause medulloblastoma. And hence, with the mutation in the patched receptor you see in Garlin syndrome, that's why those kids get medulloblastoma. Why do they get it early? Because it doesn't take very long to knock out one single allele. That's when you already have one that's mutated. So all of this biology, all of this knowledge of normal developmental biology has actually led us to understand some of the signaling pathways that occur in medulloblastoma, and it's helped us to develop not only an imaging classification, not only a classification by pathology. Now we have a molecular classification of medulloblastoma. So on a cellular level, we now study the cells that come from these tumors, and we're able to subclassify medulloblastomas into different subtypes based on what signaling pathways are disrupted. So you'll notice here that one signaling pathway, there are four molecular subtypes of medulloblastoma. And this is from Michael Taylor's paper, which is a really major paper that has come out and classified hundreds of medulloblastoma by looking at deep sequencing of all of the genes expressed and all of the mutations within the tumors, and then allowing us to categorize them by these gene expression profiles into these 4 subtypes. So remember I told you about Turcot syndrome, how the Wnt signaling pathway is disrupted? Well, the first subtype is the Wnt subtype. The second subtype has our friend sonic hedgehog involved. And the last 2, group 3 and group 4, are actually the most aggressive types of medulloblastoma. They are more frequently seen to disseminate down the spinal axis and cause those bad metastases that Dr. Scheinemann was talking about. And so those subtypes are actually the most aggressive with the worst outcome. So you'll notice that these outcomes are poor, and actually we don't really know a lot about the gene expression profiles here. We don't yet know the signaling pathways that are disrupted in the most aggressive types of medulloblastoma. And in fact, the way that we can use developmental biology as a tool to try to understand more about medulloblastoma is actually a result of putting together all the information I've shared with you already. So the scientist called Richard Gilbertson has actually developed a beautiful platform where he's actually developed a cross-species genomics model. So he's going to take a mouse model in order to model medulloblastoma safely and correlate it with all of the human data that we know about gene expression profiling. And he made some very interesting observations using this cross-species genomics platforms. So he observed that the medulloblastomas that appear to grow from the back of the brainstem, the dorsal part of the brainstem, and they're sort of very stuck there, and they're always in this classic location on MRI, they are actually caused by dysregulation of the Wnt pathway, and they tend to occur in older children. So we see that classic pattern always associated. Whereas the tumors of the sonic hedgehog group, they occur classically more within the cerebellum, where we know that sonic hedgehog is being secreted by cerebellar cells even within the normal cerebellum. And these are associated with younger patients. And so just by looking at the gene expression profiles, he was then able to model this in mouse, and he was able to actually identify the cells within the mouse dorsal brainstem in which Wnt is dysregulated. He was able to study it and discover exactly how the Wnt signaling pathway was dysregulated, and then he correlated that back to the human tumors and found a very good correlation. So this is how we can use mouse models and developmental biology to profile and study tumors and figure out the patterns of why things happen in human patients. Other ways we can do this, we can take advantage of other very elegant developmental biology approaches to discover more things about medulloblastoma. So another important thing from developmental biology is that we know that cells all originate from those very powerful stem cells that live in the floor of the fourth ventricle. Those cells give rise to other cells, which give rise to other cells, and there's all these layers in the hierarchy of normal development. So which of those cell types actually give rise to medulloblastoma? So we have these beautiful approaches in developmental biology where we can take a gene promoter that specifies only a certain type of gene. We can actually hook that up to a reporter that expresses a fluorescent protein like GFP, and we can drive the expression of an oncogene or a cancer-causing gene using only the promoter for that particular cell type. So I'll give you an example. Stem cells express a gene called nestin, and other cells that are more mature don't express nestin. So we can use the promoter from the nestin gene, hook it up to a mutation in an oncogene like P53, hook that up to a reporter, put it all into a virus, a replication incompetent human lentivirus, transfect the virus into cells, and then study exactly what kind of mutations it would take to cause a cancer to arise from a stem cell. So this paper did this paper by Robert Wexler-Rea, and he found that medulloblastoma can actually be initiated if you put a mutation of patched in front of a promoter for either a progenitor cell or a stem cell. It doesn't actually matter. You can get cancer from either cellular compartment. So another really beautiful approach we can use is using these lineage-specific promoters to figure out, does it matter what cell of origin the cancer has? And in fact, that brings me a little closer to home in terms of what I study. Why would it be important to study medulloblastoma stem cells, or those cells that occur in the floor of the fourth ventricle? Why am I even interested in studying those cells? So the very interesting thing is that we do think that those cells, stem cells, the most powerful cells that give rise to every cell within the body, and in this case every cell within the brain, those cells are particularly potent for being transformed into cancers. If you think about it, as they're proliferating and dividing, if you can somehow dysregulate them, instead of getting a normal cerebellum, you've got a big abnormal tumor. So those cells, being the most powerful cells, are also very susceptible to transformation into cancer. And so what you can see here is that in this paper, they studied human tumors and they profiled stem cell markers. And they found that, in fact, medulloblastomas do express a great deal of stem cell markers. And furthermore, the tumors that expressed the larger amount or the greater number of stem cell markers had a much worse survival in this mouse model here. So we know that those stem cell genes that are regulating the stem cell self-renewal and proliferation, that they actually may be much more potent in causing cancer. And in fact, another way of testing this is to go all the way to the embryonic stem cell level. And my colleague, Dr. Badia, who's an embryonic stem cell scientist, actually generated this very beautiful study where he took human embryonic stem cells, programmed them to go down a neural lineage to become brain cells. And then he tried to figure out, if I transform them using growth factors and media and put them into the mouse brain, what happens to them? What do they do? And the fascinating thing here is that cancer is not only dependent on the cell, but it's dependent on the environment that you put the cell into. So what he did here is he found out that when he injected the cells, the embryonic stem cells, into the skull compartment, they formed teratomas. So in other words, they just formed tumors that are made of all the different germ layers that sat there on the skull. But when he injected them into the brain itself, they formed tumors that looked exactly like medulloblastomas. So you have a very primitive stem cell that is able to give rise to a medulloblastoma if you put that stem cell into its proper environment, the brain. And so what you're really left with here is an idea with stem cell scientists in terms of trying to figure out exactly where do these tumors come from. I think that using these stem cell models has helped us to try to understand the signaling pathways and the genes that may actually cause these tumors, the medulloblastomas. And in fact, here you can see that this is a model that was published in Nature Cell Biology, where the question that we're asking here as stem cell scientists is, do medulloblastomas come from that primitive stem cell that lives in the floor of the fourth ventricle, which happens to be CD133 positive? That's a marker that I've used to characterize these cells before. Or do they come from those cerebellar granule cells that I told you about that have the receptors for the patched, which are actually CD15 positive? And in this model, in this paper, they made a really beautiful model where they said, well, you have the sort of father cells, the CD133 positive father cells at the top of the hierarchy. You've got the daughter cells, which are kind of the progenitors that originate from the top cells. And the question here is, did we get the medulloblastoma from the normal cerebellar stem cells that have made a deal with the devil to never grow old, like Dorian Gray in that movie, or from later progenitors, like the daughter cells, the CD15 positive cells, that made a deal to grow young like Benjamin Button and reacquire the stem cell properties. So that's why this paper had this title, just to show you that stem cell scientists do actually read and watch movies, and they can make literary allegories as well. And so just a different way of thinking about our work. Why do cells, are these cells actually becoming immature and regressing and reacquiring stem cell properties, or do the tumors come from the cells that were the stem cells in the first place? So the very interesting thing is, regardless of what the cell of origin is, we do know that the higher the amount of stem cell genes you have, the more aggressive your medulloblastoma. And there's many, many papers that show that stem cell gene signatures predict aggressive metastatic tumors. And so that tells us that these genes may actually be involved in causing those tumors in the first place. If we could understand more about those genes, maybe we could understand better how to block those genes from being turned on and how to prevent the tumors in the first place. So this is another very interesting paper from Michael Taylor's lab that shows that, in fact, there is evidence for a stem cell origin for these tumors. What we find is that when he did gene expression profiling of the primary tumors and then separately did a gene expression profiling of the metastasis, he found that the gene expression profiling of the metastasis is completely different and separate from the primary tumor. In other words, the metastasis did not molecularly resemble the primary tumor at all. In fact, the metastasis resembled more each other from separate patients than they did their original primary tumors. And that gives you thought again back to the original slide I showed you, where I told you that cancer is constantly evolving and changing. And in fact, you can make an analogy to the HIV situation, where we treat the virus with an antiretroviral drug, and then the virus adapts and evolves and becomes resistant to that drug, and we have to treat it with another drug. I feel very much that's what's happening in medulloblastoma. You treat the cells that are up top, but if you miss some very elusive stem cell, then that stem cell will drop down to the spinal compartment and seed a metastasis and transform into an entirely different tumor. So there's a dynamic nature to these tumors that we're not going to be able to capture unless we take advantage of the scientific models that we have. These are not things we're going to be able to discover from our patients. We have to go back to the bench and model these processes. So that's exactly what Michael Taylor discovered with this work. But the very interesting thing is that all of the metastases across the board expressed the MYC gene, both at the beginning from the top and the end. It was the only gene that was in both the primary and the metastasis. And what we know about MYC is that it's upstream of some very important stem cell genes. So the model systems I use in the lab is I study stem cells, or brain tumor initiating cells, from medulloblastomas, and we try to figure out how key developmental signaling pathways are dysregulated to modulate the stem cell self-renewal and drive tumor formation. And we want to know if genes that regulate self-renewal or proliferation of normal stem cells actually also are dysregulated in cancer. And so in my lab, we tend to look at these signaling pathways. For example, the sonic hedgehog here, we figured out in this paper that sonic hedgehog actually regulates a master regulatory stem cell gene in human medulloblastoma stem cells. And the fascinating thing is, remember that I told you about how the Purkinje cells secrete hedgehog to the external granule cells that have the patched receptor. We find that situation in normal development of the cerebellum. Well, the fascinating thing is, in our paper we discovered that there's exactly the exact same thing happening, that the cancer cells are speaking to each other in a way that resembles or recapitulates how normal cells speak to each other in the cerebellum. So it's very interesting to see the same pathways in development dysregulated in cancer. And in fact, we find that, as I told you before, we discovered that a lot of the stem cell self-renewal genes, the higher the levels of expression, are found in the most aggressive tumors. So we find that there are stem cell gene signatures that specify those very aggressive tumors, which is very hopeful, because I told you earlier that we didn't really know what genes what signaling pathways were dysregulated in the most aggressive types of medulloblastoma. So that's what the lab focuses on, to try to now pick out those stem cell genes from all of those gene expression profiles, to actually study them in detail, What we're showing here is we figured out that these two stem cell genes are very highly expressed in the aggressive, the green, the most aggressive subtypes of medulloblastoma. And in fact, we can go back to normal biology again and look at how those genes interact in normal neural stem cells and try to apply it to our cancer models. And so ultimately, what we're trying to find out here is what's the role of those key stem cell master regulatory genes? What role do they play in brain tumors? And we use all of our experimental models of stem cells to try to discover these things. So I think I'm going to finish on this note here that I brought up earlier about how now people are thinking long and hard about tumor heterogeneity. How are we going to classify the chaos of a tumor? How are we going to figure out how a tumor, which appears to be so chaotic and disruptive, how are we going to bring some order to these cells? How are we going to discover more about them? Well, we think that applying these cancer stem cell and developmental biology models will help us to discover the method behind the madness, to figure out what is the pattern, what's the answer to the puzzle, and how are we going to discover exactly what causes one tumor to become so aggressive, whereas the other responds so beautifully to therapy. So we're going to be looking for a while now to see if we can bring some order to cancer and discover the hierarchy and try to figure out exactly what cells and what signatures are expressed by the cells of origin that actually drive formation of these brain tumors. And the reason that we're focusing on all of this is once we know what those gene signatures are, and once we know what those signaling pathways are, we can target them with specific targeted therapies. So we're no longer counting on just general radiation, generalized chemo. Rather, we can find a specific readout. And so our dream would be to go back to the patient. So for example, if I had a sample of Josh's tumor, I would go back and profile his entire stem cell population of every stem cell population and gene expression profile I could find from his tumor. I would go back and read it out within his original tumor. I would find out exactly what's wrong in his signatures and target it with specific molecular therapies that would be applied in a model of having an individualized or a patient-specific therapy. So that's what we're aiming for in the future, is to add to our arsenal, to have not only me taking out the tumor, not only Dr. Scheineman meticulously prescribing chemo over months and months and mitigating all the side effects of it, not only Dr. Witten lining up every day for 6 weeks in a row to give the radiation, but ultimately to have another team in the lab working at the bench, profiling the tumor, reading out the stem cell signatures, and feeding back to Dr. Scheineman, BMI1 pathway is dysregulated, here's a molecular therapy for you. Give this pill to Josh and it'll go away. So that's what we're aiming for down the road, and the promise of this all and the reason that we're interested in this is precisely because of Josh and the Nelsons. So you guys heard a remarkable story today about a family that fought a disease that we are only hearing about today, but for them, they lived it, and it transformed them into the extraordinary people that you see here today. And it's really them that drives this work and that gives Dr. Scheineman and I the desire and the energy to carry on every day. So I want to thank them for being here today and thank all of you for being here today. And for anyone who's further interested in our research, we're doing it every day, so come and talk to us and we're happy to tell you more. So that's it. Thank you very much.